go. I believe that should be recording now.
Yep, little red lights lit up.
Thank you, sir. Okay, and where are we at? Okay, I've got 76 respondents, so I'm just going to give it a little bit longer and then I'm going to end the poll. You're all doing amazing, by the way, as always. Okay, and I'm going to end this one. So the arrow on the little picture was pointing to, correctly identified by 70 of you, the cell membrane. And as I said, it was a little bit dodged because it wasn't quite fitting on the outside. So I could see why some of you might have thought ribosome. Chloroplasts are only found in plant cells. So that would be something to watch out for. This is the animal cell and the plant cell looks different to that. Okay, I'm going to do the second one now. So what's this big red blob at the top? Question 2. And I've got to figure out how to get to it because my brain has gone funny. Aha, got it. Poll 2. And I'm going to launch that one and I'm going to move it slightly because I'm aware it's blocking the big red blobby picture. There we go. Well done to those of you who've immediately identified it, and those numbers are going up super duper quick. So what am I? Am I a palisade cell? Am I a red blood cell? Am I a sponge? Little link back to last week. Or am I a nerve cell? I did nearly put SpongeBob SquarePants, but I'm aware that caused some controversy last week, so I didn't. I'll give you a few more seconds longer. Welcome to anyone who's just tuned in. Our webinar today is on blank and osmosis. There is a reason it's blank. I shall fill that in shortly. Okay, and I'm going to stop sharing that poll now. So I'm going to end, and well done to 98% of you. That is a red blood cell. You are awesome, as I already knew. Okay, and we're going to go to the final third poll, my tricky one. So this is the hard one, and that is, what is my main adaptation? Okay, and that is a root hair cell, just in case you're not sure what the picture is. And is its main adaptation that it's got a large tail? Is it that it doesn't have a nucleus? Is it that it has a large surface area? Or is it that it has chlorophyll? Okay. And as I said, it was the tricky one, so I'm going to give a few seconds because I can see votes still coming in. Okay, going to give it 3 more seconds. Okay, I'm going to end that one now, and I'm going to share the results with you because actually, for a really tricky question, the fact that 48% of you got that it has a large surface area, that is awesome. Now, I can see why you might think it has a tail because it's got the long bit sticking out, but that bit is just making it have the large surface area. And I'm not sure if you can see on the picture with the nucleus, it's actually labelled in the bottom right. It might be slightly cut off on your screen, but little tip there is always look at the picture and see if something's on it. And chlorophyll is found in plants, and you've probably remembered its plant cell there, so well done on that one. Even though this one, it's large surface area for root hair shell. Right, so I'm going to stop sharing, I'm going to close the polls, and I'm going to move on to my next slide, still not mentioning that word. Okay, now this might look a little bit gruesome and scary, but I really like this question. Now, normally I'd get a class feedback on how this shark can find its injured prey so quickly. But the reason is to do with the top word that was missing on our slides. Okay, and what happens is the injured prey releases its blood into the sea, and then the shark can smell it. And that whole process, rightly identified by somebody in the Q&A box, is called diffusion. And hopefully you all can remember doing diffusion in chemistry with the Skittles. We put the little Skittles in the little dish, and then the colours spread out, and that was diffusion. And we're going to do a quick little recap on that after I've told you what we're actually doing today. So today's title is Diffusion and Osmosis, and we are going to look at what diffusion and osmosis are. So a recap of diffusion, and then I'm introducing you to osmosis. And then we've got how substances actually move into the cells, and looking at some cells, and then conditions and organisms and plant and animal cells and what actually happens when these things occur in them. Okay, so just to remind you, the Q&A, not the Q&A, the hand up thing doesn't actually do anything. So if you have a question, pop it in the box, and Miss McKinley and Mr. Lamb will answer any questions you have or let me know about them. Okay, so I am going to move it on. So this is my reminder. So diffusion is the movement of particles along a concentration gradient, and they go from a high concentration here to a low concentration where there's no particles, and they spread out. Okay, and this can happen in gases and it can happen in liquids. Now, if you're in my class, you should be able to remember me teaching diffusion with the anecdote that somebody had a dodgy curry the night before, and unfortunately they've got bad wind, and if you're sitting next to them, you're going to smell it first, and then it's going to take a while and spread out and the whole class will smell it. That's diffusion. You have the high concentration of smell where the person who had the dodgy Indian is, and then it spreads out and everyone can smell it. Okay, so diffusion is the movement of particles from an area of high concentration to an area of low concentration across a gradient, a concentration gradient, and that's the fancy words that we like to use in science for that. Okay, I'm still talking about it. So again, I've got a different picture here just because I like to use different images. So we've got high concentration particles here. This could also be an aerosol, perfume, room spray. When you spray it, it's highly concentrated in one area, and then over time it spreads out to where there's a low concentration. And then some key bits for you, just so you've got them. There's my high concentration to low concentration again. And it's random. It means we can't control it. So we don't know which particles are going to move where and bounce into what. It's completely random, and it just occurs naturally. And then the one that's going to be important later on, if I can get it to click on, is that it's passive. Okay, and this means you don't have to give it any energy. So you don't have to do anything when you spray the aerosol, it just happens. No energy is needed to be put in. Diffusion just takes place. Particles will go from where there's lots of them and spread out in a container or in a room where you have smells. Okay, hopefully that is a nice recap for you of diffusion. Okay, so here is linking in with our biology because we've been doing cells. Okay, so this is diffusion in a normal cell. So we've got our animal cell, and you can see just visually by looking at it, we've got a high concentration of particles outside the cell and a low concentration inside the cell. And then over time, my bad, I just clicked the wrong bit. They balance out and you end up with an equal concentration inside and out of the side of the cell. So diffusion will still happen here. They'll still bounce in and out, but they've equaled out. So it's gone from high to low, and then we end up with a balancing out. And that happens in cells so that we can get substances like glucose and oxygen into the cells, and we can get some other things out of the cells. Now, my next slide, I've put some polls on, and I'm just going to talk you through because this is quite tricksy. Okay, so we've got this lovely picture at the top here, and we've got proteins, okay, represented by the diamonds. We've got oxygen represented by the squares. We've got glucose represented by stars, and we've got carbon dioxide represented by these oval shapes. Now, the key thing that I need to tell you, because I'm going to ask you these two questions in a minute, which one will move into the cell? Obviously, can't answer the why, and which one will move out. The key thing I need to point out is, one, you can count the shapes. So you should be able to work out where there's a high concentration and a low concentration. If we're moving into the cell, we're going to go from high to low. And the other thing I need to point out is that proteins, which are the diamonds, are super, super, super big. So actually, these little diamonds won't fit into the cell. Okay, so I'm giving you that now because it's tricky otherwise. And I could see, if I was looking at protein, I'd go, well, there's one on the inside and 3 on the outside. Protein will move in, but it won't because it's too big. Okay, so I'm going to do my little poll just to see who's been listening. Okay, and we hopefully will see that we're all able to count, hopefully. So I'm going to try and move it out of the way. I'm moving it hopefully over here. Okay, hopefully that means it's out of the way and you can see the picture. So we've got the little diamonds are the proteins, we've got little squares are oxygens, we've got the little stars are glucose, okay, and then we've got carbon dioxide is the ovals. So which one do you think will move into the cell? And welcome to anyone who's joined us. I've just noticed the numbers are slightly higher. You're all doing really, really well, by the way. I'm super impressed because this question is really tricky. I'm just giving you loads of information there and gone, answer the question. I'm going to end up meowing in a minute just while I wait for a few more. Meow, 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 meow. I should probably go pika, 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 but I like meowing. Okay, numbers are slowing off a little bit, so I am going to end the poll and I'm going to share the results with you. Okay, so, hopefully you can all see there that the majority of you correctly identified that it is going to be oxygen. So well done to you guys. If you look at the oxygen numbers, just to say why, we've got 2 oxygen inside the cell. And we've got 1, 2, 3, 4 on the outside. So that means there's a higher concentration outside than inside. So the oxygen is going to move into the cell. For those people that put protein, you're right in terms of concentration, but the protein's too big, so it wouldn't get in. And then glucose will diffuse, but not as readily as the oxygen. So there's 3 on the outside too, so I'd accept that one. And then carbon dioxide is 1, 2, 3, 4, 5 on the inside and 1 on the outside. So there may be diffusion happening there, but it won't be into the cell. Okay, I'm going to stop showing results. And hopefully, because of what we've just discussed, you'll be able to tell me which one of those 4 goes out of the cell. Okay, so I'm going to launch it. There we go. And I'm going to move it out the way again. So which one is going to go out of the cell? I'm going to give you a few seconds. The options, by the way, are the same ones. So protein, oxygen, glucose, and carbon dioxide. You can tell I don't like awkward silences, can't you? Okay, and I'm going to end that one in 3, 2, 1. And there we go. And I'm going to share because you're all listening and 82% of you recognised and counted or followed along with my counting that carbon dioxide is going to move out. So again, just to show you 1, 2, 3, 4, 5 inside the cell and 1 on the outside, that carbon dioxide is going to move out. And this is something that actually happens in our cells. So we want oxygen in so that we can respire, and we want to get rid of the carbon dioxide because it's dangerous to us and we breathe it out, which is something you look at later on, but I'm just filling it in. Okay. Right, this on the board is osmosis. Now, osmosis is similar to diffusion, but there are 2 main key differences. And what I don't want anyone here to do is say osmosis is diffusion, because it's not, okay? So try to avoid doing that one, okay? So osmosis is the movement of water, okay? So diffusion can be the movement of smells and dyes and anything that can move that's in a liquid or a gas, but osmosis is purely about water, okay? It follows the same principle, so it's from an area of high concentration to an area of low concentration, and it's the movement of water. And the key other thing with it is it's got a semi-permeable membrane. Okay, so the full definition of osmosis is the movement of water from an area of high concentration to an area of low concentration across a semi-permeable membrane. There we go. That is osmosis. Now, I've got another little slide with another picture just to sort of show you again. Now this, where it gets slightly more complicated in that we've got a sugar solution, okay, and I'm not going to go into this too much, But essentially, when you've got sugar solutions, it changes the amount of water that there will be, and it can alter it slightly. And I've put a little video link on my next slide of a song, which will help you with that if you want to look at it in a bit more detail. But again, movement of particles from an area of high concentration to an area of low concentration across this semi-permeable membrane. And that just means the membrane's got holes in it. If you think we talked about the cell membrane, it's got the holes for things to go in and out. Semi-permeable just means it's got little gaps in it so the water can move. The glucose, you should be able to see this little glucose bit here, it's too big. Okay, glucose never moves. Okay, and a lot of things with osmosis will talk about glucose and water, but just remember, key tip, glucose never moves, only the water moves. Right, and then I found this little definition, and I put this on, not quite the same as my one, but I liked it because it told you that osmosis comes from the Greek word meaning push. I thought that was an interesting fact to share. Okay, and it's the movement of water molecules, there we go, through the membrane from an area of high to low water potential on this one, but high to low concentration. And very, very simply, that is what osmosis is. Now I want to make things a little bit more interesting in a minute. I thought it was the next slide. It's not. Bear with me. So osmosis happens in plants and animal cells. Okay, and it's a really, really important thing. Okay, so with your plants, especially at the moment, I know mine are getting a bit wilty because it's very hot. When they lose water, they start to wilt, and you can see it's because they start to lose their shape. Okay, they start becoming flaccid or plasmolyzed, and that just means they lose their shape, and it's when their leaves get all droopy and you realise you've got to water your plant. On the other side of it, if they have lots of water, they end up with a turgid shape like this, and that keeps the plant upright. So water is really, really important in plants. Okay, and when they wilt, they keep all the bits together, and then when they get water, they can go upright. The thing that helps with that is the cell wall. Okay, so the cell wall means plants, if they lose water, they can keep all the bits together. Okay, you can see it's not damaged the cell so much, and it keeps their shape. Hopefully that all made sense. Right, and just because I like to show you, this is what happens in onion cells. If they lose their water, you can see the shapes have got all wiggly and they've lost their sort of defined shape. And then we've also got, coming up, there we go, onion cells. So same cells, full of water, they've got their nice big square shapes. There we go. Right, animal cells. Okay, so animal cells, they don't have that cell wall. Okay, so that means that two things will happen to them. They will either, if they get too much water, swell and burst, or if they don't get enough, they will shrink. Okay, and I'm going to ignore my little question at the bottom. If you want to answer that in your classroom, you'll be able to. Okay, here's my little picture then. So we've got our red blood cells, and if you place red blood cells in water, they are going to take that water in by osmosis, and then they'll burst. Or if you place them in salt solution, because the salt solution is going to be a lower concentration of water, the water would leave, okay, and then the cells would shrink and shrivel, and then they wouldn't be able to work, and that would be really, really dangerous for you because they can't carry that oxygen around. Okay, so that's the main difference. In plants, it helps them keep their structure and they can cope with a bit of wilting. In animals, they'll either swell and burst or they'll shrink and shrivel. So for animals, it's really, really important that we keep our water balances correct. So where they say keep hydrated and make sure you drink enough water and things like that. Right, now, these are gummy bears, and I was having a big discussion last night about if you can get gummy bears in the UK, and I'm not sure you can. Essentially, they're little sweets with gelatine in, okay, and someone did an experiment with them, and this is the original gummy bear, and they put them into some salt water, and you can see, hopefully, that osmosis has happened, and the water has left the gummy bears. Okay, they also then put them into sugar water, and they've gained shape a little bit, and then they've put them into regular water where they've expanded and they've taken that water in. So gummy bears have got a semi-permeable membrane, okay, and that means that the water can either go into the cell like here, or it can leave the cell here. Okay, now in the bottom of this slide, when it goes into your classroom, and I don't want to get in trouble with Mr. Lamb for this, I've put 2 links. One is to the gummy bear song, okay, which is quite good. It gives you the information. It just does that osmosis is H2O diffusing, which I don't like. I don't want you using the word diffusion for osmosis, so I'm putting it out there. But I've also put a link to a little experiment to do with gummy bears. Now, I'm not saying everyone should go out and run out and ask parents for gummy bears, because one, I wouldn't know where you get gummy bears, okay? But if it's something your parents agree to, then you could have a try with some sweets you might have knocking around, have a play, because I'm hoping most of you have got water in a tap. Okay, so if you want to have a look, it's in the link, but there is no demand on anyone to do this, okay? And if parents say, no, you can't have a look at anything like this, then, you know, listen to parents. Right, moving on. I think, oh no, I have a poll to do with this. Right, so I'm going to put the poll up. And here we go. Okay, so my poll to do my gummy bear picture was, which set of gummy bears have experienced osmosis into them? Okay, so is it the saltwater gummy bears? So osmosis into them, movement of water into them. Is it the original gummy bear? Is it the sugar water gummy bear? Or is it the regular water gummy bear? I'm going to give you a few minutes to answer that one.
Hello, Miss. While they're doing that poll, certainly in this country, Haribo gold bears work just as effectively as gummy bears, and you can get those in most supermarkets.
All right, thank you, sir. Oh, we were trying to think about things like cola bottles or Haribo eggs last night. We weren't sure.
I think they would all work.
But as I said, there's no pressure. It's just there for a little bit of fun if you fancy it. Okay, I'm going to give you a few more seconds on my little gummy bear question. I now want to start singing, oh, I'm a gummy bear. Oh, I'm a gummy bear. That's all you're getting from me, though. I really can't sing. Hopefully that will be stuck in your heads, though, and you'll go away going, I'm a gummy bear. It's a good song if you haven't heard it. Right, anyway, I'm going to end the poll. And 60% of you recognise that regular water. So the key here was osmosis into them. So the movement of water into them. And the biggest tip I can give you is, oh, I wanted to share it so you could see the results, is that the regular water ones have got larger. So it shows that they've gone from the little size up to this big, massive size. And that means the water's moved into them, okay, across that gradient from a high concentration outside the gummy bear to a low concentration inside the gummy bear. Okay, so we've gone from high to low, and then that results in the gummy bears getting bigger. Salt water, osmosis has happened, but it's the other way. So out of the gummy bear into the water. So the water's left the gummy bears, which is why they've shrunk up. Okay, right, there we go. So that is essentially osmosis and diffusion. And I've put this in, and again, I've used my tiger slides. So at the very basics, everybody in year 7, hopefully, should, if they listen to this and look back at the slides, be able to define diffusion and osmosis. So everybody should be able to do my little kitten task. What is diffusion? What is osmosis? And then I've put for the tiger tasks, find or draw a picture, up to you, of diffusion and describe what's happening. So what is going on with those particles? So it's a bit more of a detailed explanation and description of what diffusion is, and the same for osmosis. Okay, and then we've got our challenge at the bottom, which is our lion, and that is comparing the two. So how are they the same? How are they different? And because I've realised I've missed a bit, I'm also going to tell you that osmosis is passive, so it doesn't require energy, and it is also random. So the particles move randomly. Needed to put that in there just to make sure I've covered all the bases. So you need to be able to compare them both. How are they the same? How are they different? And then my bottom question was compare and contrast the effects of osmosis in animals and plants. So that's that little bit I did about animals and plant cells. Now, this is not quite the end because I've added an extension task on. Okay, so this is the main bit. And then for those of you who are super duper awesome, I have added in an extension about this little cell, because we've actually done everything about cells apart from these guys. Now, this is Euglena. Okay, it is a single-celled organism. That means this is it. This is what makes it up. Okay, and they are found in ponds and puddles, and I've put a little video clip link there for your lessons if you want to watch a video later of what they look like. Okay, now they are super duper interesting because they have parts like a flagellum, which plants and animals don't have, and then they've got the nucleus, and then they've got chloroplast, which make them seem planty, and then they've got a cell membrane, but there's no cell wall there, so they're very, very interesting. Okay, this is what they look like in real life, and I think they're pretty cool. Okay, and the video shows you them moving around and things. I think they're super nifty. And what I've done as an extension is I've added this. So your job as extension would be to tell me, are Euglena animals or plants? And I've put some evidence about them, so facts that are known, into this document. So there is evidence that suggests Euglena is a plant. There is also some evidence that suggests it's an animal. There is some evidence that's going to suggest it could be both, and there is evidence that it might not even be either of them. So these are the cards, and I've done 2 of them for you. I've colour-coded it, so I've made it nice and easy. So I thought, well, it doesn't have a cell wall. That would make it an animal. But it's got a vacuole, so that would make it a plant. And essentially, your extra challenge job, your extension task, would be to go through these and maybe change them different colours. So if you think it's animal, then you make it red, and then if it's plant, green, and then if it's both, you could do a different colour, and if it's neither, then you could put it a different colour, and then write a little description afterwards just to say what you think. Okay, so is it a plant, is it an animal, is it neither, is it both? And as I said, there is evidence to suggest it could be all of those things, and I want to prove a point to you all right now, so I'm going to launch a poll. Okay, so I'm launching it. From what I've said, do you think it is a plant, an animal, both or neither? Okay, so this is just me seeing if you're having a listen. Because, you know, I'll just move it over so you can see the picture. Oh, bear with me. Why is the poll not moving? Oops, hang on, sorry. There we go. Can't get the poll to move for some reason. Oh, there we go. Okay, going to give you a few more seconds. Do not worry about your answer. I can see some of you are not going as quickly. I mean, it's got a flagellum. What could that mean? But it has got chloroplast. What could that mean? Going to give you a few more seconds. Doopy 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 doo. I'm a gummy bear. Oh, I'm a gummy bear. Don't worry, I'm not going to go on X Factor. Okay, I'm going to end that there, and I'm actually going to share your results. Now, the reason I did that poll is because what I want you to get from this is there's no right or wrong answer. Okay, you could argue that Euglena is a plant cell. You could argue that it is an animal cell. You could argue that it is both, and you could argue that it's neither. Okay, the key thing with this end task is to say why. So if you said, I think it's a plant cell, you would have to back that up with Euglena is a plant cell because it contains chloroplasts. And then that is justifying your answer. If you thought animal, it's an animal cell because it has a nucleus and it doesn't have a cell wall. It's both. You could argue it's got chloroplasts and it has a cell membrane, that sort of idea. I basically just wanted to show you with this that there's no right or wrong answer as long as you justify your answers. Okay, I don't know if I shared it with you, but there you go. My key thing, as I was saying, was that there's no right or wrong with it as long as you can justify. Right, and I've got a few more minutes. So, and this came from Mr. Collins because we're trying to get some different scientists around the world in, and I've put a web link in on this one. So last week I was trying to find somebody, and this is Ada E, and I'm probably going to pronounce names wrong, which is really bad on me, Yonath, and she got the Nobel Prize in 2009 for chemistry for her work on ribosomes. Okay, and last week we did a bit about ribosomes and how they make proteins. She was born in 1939 in Jerusalem, and in a part which is British Mandated Palestine, which is now Israel. Her life is really, really interesting. Now, I'm not going to talk about it too much. The link is there, but I just wanted to include some different scientists and different people for you to have a look at, who've done amazing work. So she did amazing work on ribosomes, and her life is pretty interesting and inspiring. Okay, so I just wanted to include that in there for you. Now, I'm actually pretty much done. So I'm going to move back to that awesome picture of Euglena, and I'm going to say, Sir and Miss McKinley, do we have any questions in the Q&A box? And if you have any questions, put them in the Q&A box. Doobie 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 doo.
Hello, Miss. Well, I've got a couple of things. First of all, at the beginning, lots of people said nice hat, so I thought I'd share that with you.
Thank you.
And also a lot of people have said thank you, so I thought I'd share that with you as well. I've got a couple of questions. Some people have said, where do they find the video and the slides after the lesson?
So the slides will all go into the Google Classroom, and then for this one, if you just click on the clip, I'll show you. It should load you straight onto the little video. Okay. Now I've got to work out how to get back from that, so bear with me. Right, hang on. No, there we go. And on the other ones, if I hit escape, there we go. So you can click on this one. You can see at the bottom, there is a web link. So that's to do with the gummy bear experiment. And then one of them somewhere, where did I put it, is the video link for the gummy bear song. It may very well be on this one, just further down. But I will double check that one. It's definitely on one of them. But all the links I put in the little speaker notes underneath.
Brilliant. And the recording of the webinar, if you want to watch it again, should be in your Google Classrooms as well, but it won't be until later on today.
Yeah, not till later today because we have to upload it. I am going to stop the recording now, but feel free to add more questions. So, there we go.